Cal, and yeah, so just a heads up, this meeting will be recorded, and so if anyone is uncomfortable turning their cameras on, it's totally fine. And so this panel is part of the Spectrum's Fall 2020 Acceptance Week, and we are very honored to have 3 professors joining us. And I think one of the so the 3 panelists we'll have will be Professor Stephen Hinshaw from Department of Psychology, Professor Laura Starponi from Graduate School of Education, and Professor Dan Feldman from MCB Neurobiology. And I think Professor Stephen Hinshaw, he is now in an inpatient surgery, so he may not be able to come to this panel, but I will provide his contact info to anyone who is interested in his research and if anyone has any questions. And so every panelist will take about 20 minutes to go over their study and share their research regarding autism. And as you can see, every professor is doing like very different research regarding autism. So we're very glad to hear about like different fields regarding autism. So, and at the end of this panel, we will have a Q&A session for anyone who has questions. So before we get started, I just wanted to provide a brief introduction to each of our panelists. So Professor Stephen Hinshaw, as I said, he is a professor of psychology, and he is a professor of psychiatry and behavioral sciences and vice chair of child and adolescent psychology at UCSF. And he is the PI of Hinshaw Lab, and his research focus is on stigma in relation to mental and neurodevelopmental conditions. And as he is not able to join this panel in person, I have listed his email right here. If anyone has any questions about like his research, just feel free to email him about like any questions you may have. And our second panelist, Professor Laura Starponi, who is already here with us in this panel. So Professor Starponi, hello, is an associate professor of language and literacy in the Graduate School of Education with a research focus on language and literacy practices in children's development and education. So with relation to autism, she is studying about language practices in autism. Yeah, so our last panelist is Professor Dan Feldman, who is a professor of neurobiology here in the MCB department, and also the Coates Family Indulge Chair. And he has a research focus on the neural circuit function and sensory processing in the cerebral cortex. And in relation to autism, he is studying about the neural circuit basis of autism with transgenic mouse models. So yeah, I hope this provides a brief introduction for all of our participants. And now I will leave the table to our first panelist, who is Professor Laura Starponi. Professor Starponi, if you want to screen share, I can stop sharing right now.
Yes, please. I would like to share some slides.
Yeah, for sure.
So let me see if I. Okay, can you see now my slides?
Yes.
Okay, and you can hear me, right?
Yes.
Okay, we are set. Well, thank you very much for inviting me to this panel. It is both a privilege and a pleasure to participate to a scholarly meeting in which I can assume that participants are familiar with the notion of neurodiversity. In fact, I know that a good number of you have contributed a great deal to advocating neurodiversity. So it is an honor for me to have you in my audience today. Okay, let me see how to move forward. Today I would like to briefly characterize the overall scope of my work on autism before zooming into the specific focus of today's presentation. My background is in developmental psychology and interactional linguistics. Over more than a decade now, I have developed a program of research on autistic language that has augmented traditional scholarship on autism in two main ways. One, by discerning dimensions of communicative competence in autistic children that were previously undocumented and largely unrecognized. And two, by identifying interactional processes that have a bearing on the manifestation of autistic language. In a series of qualitative and mixed method studies, I have examined prototypical features of autistic language: echolalia, pronoun reversal and avoidance, and pragmatic deficit. These are the traditional labels, to be clear, which I resignify or debunk rather than confirming or endorsing. By focusing on how such language phenomena manifest in the speech of autistic children as they engage in spontaneously occurring verbal activities in everyday context, my work complements traditional psychological research, which is primarily carried out in laboratory settings. In fact, my work advances interpretations of these phenomena that complexify the classic view. In mainstream autism research, these verbal behaviors are generally taken to be symptomatic of an underlying condition, taken as direct manifestations of deficits in areas such as social relatedness, perspective taking, and cognitive flexibility. While not denying that there is a neurological basis of autism, and I really look forward to Dr. Feldman's presentation later, and while not denying that autistic individuals have difficulties in certain domains of functioning, my work provides caution and alternative interpretations, leading to insights that have the potential to transform our perspective on language in autism, promoting a view that is well aligned with neurodiversity scholarship and pronouncements. As an example, today I'm going to focus on one of the most salient features of autism according to the biomedical characterization, the DSM-5, to be more explicit. The feature that I want to focus on is repetitive patterns of behavior. And I will consider specifically ritualized patterns of verbal language, which most frequently are referred to as echolalia and formulaic utterances. Until recently, such perseverative behaviors have been interpreted as automatic responses, often disruptive of social interaction, with no or minimal communicative function. Neurodiversity activists and scholars, on the other hand, have pushed to reframing repetitive behaviors as semiotic phenomena, that is, meaningful sensory explorations and forms of self-expression. In this presentation, I leverage methods from interactional linguistics to contribute further insights into repetitive verbal behavior in autism. My data set for this study was generated from spontaneously occurring verbal interactions of 10 autistic children aged 5 to 7, interacting with their family members as they engaged in habitual activities, primarily, but not only, in home settings. These activities were video recorded bi-weekly for a month, and a total of approximately 16 hours of video recording for each child were obtained. In this presentation, I will present video extracts selected to give a most clear illustration of the general findings of my analysis. As for my method, my first methodological intervention is to identify every instance of repetitive verbal behavior. I thus assemble a collection of all echolalic and formulaic utterances in the data corpus. I then consider a heuristic question that is central to any interactional linguistics inquiry: the question of why that now? This question is analytically fruitful in that it brings us to consider the sequential context in which focal behaviors, such as echolalic utterances, occur, rather than connecting this behavior solely to an underlying disorder. So it's not simply because one is autistic that repeats utterances, but perhaps it's also because of things that happen before, or things that are set in motion with echolalic utterances. We'll see that. So this analytic procedure brings to light properties and functions of autistic repetitive language that previously remained unrecognized. And I will discuss and illustrate today three main findings. One is that even the most functionally opaque and seemingly autonomic form of echolalia is synchronized to surrounding talk. So it's not random or automatic, but takes into account what's going on around the child who's producing these echoes. Two, formulaic sequences are not monological strings uttered all at once, but are developed conjointly with the interlocutors. So they're more kind of co-constructed than automatically regurgitated by the child. And three, echolalia and formulaic utterances can be mobilized to resist and divert these preferred courses of action. So displaying great agency and control of interaction, or an attempt to do that. Now to illustrations via examples. In the first two extracts, we observe Florina. These are pseudonyms. You'll hear actually the actual name uttered in the video. Florina is a 6-year-old autistic girl whose talk is largely made up of echoic utterances. Florina is an only child brought up almost exclusively by her father. She's very affectionate and likes to be with her father when he hangs out with friends, like in this occasion. We see Florina here, Dad, who's holding her, and a close friend of Dad on the left of the screen, and the friend's daughter, who is out of the camera on the right, who does not know Florina as much, not as much and as well as Dad and his friend. Here they are in the back patio of a pub, eating pizza and drinking beer, the adults. Florina's speech is highly echolalic. In this first extract, we observe Florina utter a delayed echo, only partially intelligible, despite it being uttered in high volume. As you watch the video segment, please pay particular attention to Florina click sounds, 2 of them which precede every time the delayed echo. Now I'm going to play the clip, and then I'm going to kind of offer further analysis of the clip.
But you gotta ask. No, please, I gotta ask just for a little while. Sure enough, she actually he's against the button. She's gonna jump with her. She's heartbroken, you know. Hi.
Hey, here's your salad.
Oh, thank you.
What's up? Can we eat? Do you want to eat some salad?
She'll have some bread.
Okay.
Okay, here's the transcript. So Dad's friend is telling a story. It is right after he reaches the punchline that Florina produces a delayed echoes. Here you can see them in line 9, 11, and 12. And the vertical lines are transcription conventions for the click sounds that she produces. So Florina's clicks and delayed echoes are not produced in the middle of the speaker's sentences, but occur right after he reached the punchline. So they're not perceived as interrupting neither friend nor Dad. At the same time, Florina's speech here does not seem to be oriented to solicit an uptake, not to be really opening her own line of conversation. And this is true for the second delayed echo as well in line 26, which is itself prefaced by 2 clicks in line 25. Neither Dad nor Dad's friend, who know Florina as well, treat Florina's click prefaced echoes as communicative. The friend's daughter, who is not familiar with Florina and her speech repertoire, does, however, though minimally, and Florina does not follow up with it. Now consider a second extract from shortly after in the same event with Florina. Dad's friend leaves the table to reach the restroom. Florina does not seem to notice it initially, as she's focused on eating her pizza, removing actually mushrooms from it. When she seems to realize that Dad's friend is gone, she immediately produces another delayed echo. Notice that this time the clicks are absent and she seems to be more oriented towards the interlocutors. I'm gonna play the clip now.
Who came with the free beer and free pizza?
Oh, you don't know there is a back.
Oh, right.
There we go. We go for that.
Oh, she's taking it from that way.
Yeah, she doesn't like mushrooms.
Yeah.
It's set to go. It's just to go. Okay.
So here we observe Florina producing echoes 3 times. It seems that Florina's echo is a commentary on Dad's friend's momentary absence. Her utterances are not prefaced by click and seem to be oriented more clearly towards the co-present participants. Indeed, Florina repeats her echoic utterances 3 times. That is until she receives an uptake from the friend in line 12. At that point, she doesn't continue repeating her utterance. While Dad does not proffer a response to Florina, he bends over her in a gesture that seems to be doing recipiency, so to be attending to her communicative initiative in line 8. So very different ways of using echolalic utterances in this girl. One prefaced by the clicks, not oriented towards interaction, but nevertheless produced at a point that is sensitive to the talk that was ongoing around her. And the second one, more clearly positioned and packaged to solicit a response, to be more communicative with respect to the other interlocutors. Let's move to another example, another child. In this third extract, we observe Aaron, a 6-year-old child with his mom. Aaron has a more developed language, highly repetitive, but also other generative sentences. Here we see the child at the beginning of a bath, a favorite moment of his days. And mom is helping him regulate the temperature of the water. We will see that repetitions here in this segment are mutual between mom and Aaron, and that Aaron builds on mom's prior turns to produce generative variations rather than exact echoes.
Okay, now put the plate in. There we go. It doesn't get too hot. Here we go. You don't want it to be hot, believe me, or else it hurts. It hurts. Oh, it hurts. If it's too hot, it will burn you. It is not
got cut actually. Anyway, let's move. So Aaron echoes Mom's warning that it's gonna be hot. And then Mom deploys "or else" as follow-up to her warning in line 7. Or else is a discourse marker that Aaron frequently uses. As a matter of fact, he utters "or else" almost in unison with Mom in line 8. Then Mom cautions her son about getting hurt as the possible consequence of letting the hot water run. Drawing on Mom's prior turn, Aaron formulates a double generalization. Hot hurts. All hot hurt in line 11. Then Mother again picks up an element of Aaron's talk, that is hot, and offers an expansion, rather general itself, in line 12. And Aaron then builds another generalization based on it. Hot water burns in line 13, which brings the sequence to its close. So we see that, and in line 15, sorry. So we see that leveraging conversational material produced by his interlocutor previously in the sequence, Aaron is able to use norm formulations, abstractions, and generalizations as progressivity resources that exercise considerable conversational control and bring the sequence to its close, having the last word on the matter. A final extract. In this one, we observe Aaron again once the bath is over. He wished actually to stay in the bathtub longer and play more, but Mom is trying to move him to prepare for bed and sleep. We see that Aaron resorts to echoes and formulaic language to resist Mom's desired course of action.
Shriveled. It's shriveled. Oh, your fingers? Let's have a look. Oh my goodness. You got shriveled like a little raisin. Yeah. You got shriveled like a raisin.
If you don't.
If you don't, what?
Take a bath.
If you don't take a bath, what?
In 2 hours.
Here, Adam.
I'm gonna do
Adam,
all
if-
day long.
Adam, look. Put these on with the crotch in the front.
Yeah. Catch.
Crotch in the front.
Catch. Is it crotch day?
Is it crotch day? I suspect it is.
Catch.
Now, Adam,
After
That's a good
December
idea.
comes January. After August comes July.
So Aaron is still in the playful mood from bath time and tries to recruit Mom again in co-construction of sentences. Mom, however, seems to have some urgency in bringing Aaron to be ready for bed. When Mom interrupts Aaron's play and delivers him instructions in serious flat tone in line 18, Aaron begins to utter repeatedly "catch" in line 19 and following, and then shifts to some formulaic sentences on the sequence of month, diverting and resisting Mom's projected course of action, which clearly is not his favorite at that moment in time. To conclude, in this presentation, I hope to have articulated a compelling argument for complicating the traditional view of repetitive verbal behavior in autism, providing evidence that support neurodiversity claims on the functionality and meaning of perseverative phenomena. A close analysis of the composition of autistic utterances and their position in sequential context has brought to light the complexity and interactional significance of autistic repetitive speech. The first two extracts revealed that the talk of an autistic child whose verbal utterances are overwhelmingly made up of echoes is highly sensitive to the interactional circumstances. In addition, we saw that the echoic utterances are packaged in a way that marks them differently, as either oriented to self or inviting an uptake from the co-participants. As such, echoes can be said to include a meta-communicative marker, a signal that indicates how those echoes should be interpreted. In the third extract, we saw the production of formulaic strings that are often co-constructed by the child with autism and the interlocutors, and present a considerable degree of variation. Thus, rather than solely being a manifestation of an underlying proclivity to repeat, formulaic language also emerges as resources that the child mobilizes to exert agency and display competency in the unfolding interaction. The final extract has illustrated how repetitive language can be mobilized to divert or resist these preferred courses of action. Also, as such, repetitive language is interactionally oriented. So repetitive verbal behavior as a form of autistic semiosis communicates and meta-communicates. So we are better off listening attentively to things that autistic children say again and again. Thank you. I'm done. I'm going to stop sharing the screen now.
Yeah, thank you so much, Professor Starfoni. It's definitely, we learned definitely a lot in this past 20 minutes about echolalia and repetitive language, and those extracts. I don't know, personally, I found them really inspiring, and along with your analysis, I think they really enhance our understanding in this short 20 minutes about like autistic language and behaviors. So yeah. So thank you again. And so now let's hear more about like the biological side of autism. So let's turn the table to Professor Feldman.
Okay, great, thank you. That was really fascinating. I have to say that is a side of understanding behavior in autism that I don't normally think about in my work, but you know, when you work on animal model systems, like I'll tell you about in a second, you tend to think about underlying mechanisms that can explain differences in behavior rather than thinking about the motivation and the utility of the behavior itself. And so I think that was really quite interesting. Okay, so I wanted to give you guys a different perspective, I guess. I don't have a real research talk to give. I have a few slides to kind of illustrate the sort of approaches that we take. But I thought it would be useful, hopefully for you guys, to hear about how people study biologically the differences in brains of autism model animals, right? We don't study autistic subjects themselves, and how we approach that problem. And actually, I'm also really interested in hearing some impressions or feedback from you guys about your approach to this, because I really come at this really from the biological side. So I will also share a few slides here. Okay, so from a biological point of view, one of the most interesting discoveries about autism in the last decade or so has been that there's a much stronger genetic component than I think people had suspected recently. And there's been you may be familiar with this, there's been a big effort to identify genes in humans that confer some risk for autism. Now, this is not a disorder where there's like, you know, a genetic mutation that causes a disease. As you know, it's a complicated spectrum of disorders. I think neurodiversity is a really great way to think about it. There's many, many people who are somewhere on the spectrum that you wouldn't really say are impaired in any way, right? They're different in many ways. But then there are, you know, many portions of the spectrum, perhaps you might think of it that way, where there is severe effects on behavior and on communication, often also on sensory dysfunction is another aspect of it as well. And so how do we understand how genetic causes might contribute to some of this? And I have to say, there have been many, many studies of this recently. There are about 200 clearly identified genes in humans that are what are called high confidence risk genes for autism. That means that they have statistically clear and repeatable association with the disorder across many different groups of subjects, of families. Now, interestingly, these genes don't all strike the same cellular pathways or even the same, general functions within the brain. So there are many different types of genes doing many different things in cells. How is it that these very different genes end up converging on something that we would call in common, a diagnosis of being on the autism spectrum, even though autism is not one thing? So this is one question that a lot of neurobiologists are very interested in. Can we use animal models of these genetic mutations in mice, usually, to try to figure out, is there something that occurs in common that's different in the processing of these brains of mice that might explain something about how these genes are related to this similar kind of condition? And this is what I'm trying to illustrate here on the left. So each of these dots is schematically illustrating, one of those many, many different gene mutations which are associated with autism. They're hitting different functions of cells, but how could they in common affect behavior or social communication or perception? One possibility has been proposed at the neural circuit level. And here you guys may know that as information moves around in the brain is processing that information. That is really the basis for all of our sensory and motor and cognitive functions. And a lot of our thinking about how that works boils down to a very simple idea, which is over here on the right. This is a neuron, a pyramidal cell in cortex. And we know that neurons receive both excitatory signals from other neurons, and they also receive inhibitory signals from other neurons. These two types of signals will either excite this cell, we call this depolarization of the cell, or they'll inhibit the cell, which hyperpolarizes the cell. This is actually moving the membrane potential of these neurons in opposite directions. And we know that neurons are in the job of integrating excitation and inhibition. And it's something about the interaction of excitation and inhibition which determines how this cell generates its signals, which are action potentials or spikes. And that's what conveys information in the nervous system. So the main idea that people have come up with over the last 10 years on the neural circuit basis for how these different gene mutations might all contribute to something in common about brain function is that maybe they all change how excitation and inhibition are integrated in the neurons. And the most common hypothesis is that they change the ratio of excitation to inhibition. And specifically, people have argued either that they decrease inhibition in the brain or that they increase excitation in the brain so that the ratio of excitation to inhibition goes up. And you might imagine that if a cell is in the business of integrating E and I to decide when to spike, if the EI ratio goes up, this cell might produce more spikes than normal. Okay, now that is actually a very interesting proposal, that cells might produce more spikes than normal, because there's a strong association in about 12 or 15% of cases across many different forms of autism with seizures and epilepsy. And of course, seizures and epilepsy are too many spikes, right? So maybe this could also explain some of the reason that some forms of autism are associated with epilepsy or seizures. And even if those excess spikes don't cause epilepsy, maybe those excess spikes represent some kind of noise in the computations that neural circuits are doing. And this could explain the cognitive or the sensory issues that many different subjects have. So this is the general idea, and this is just one hypothesis, but it's now a concrete hypothesis that you can study neurobiologically to ask whether it might be true. And I'll tell you some of the approaches we take. I'm actually not going to go through any data that we have, but I can tell you about our general findings at the end. But let me just say a word about why, it seems attractive to study this kind of idea. if there are some individuals with autism or some families, family members with autism where, people are affected seriously enough that they're looking for some kind of medical treatment, it would be a very difficult endeavor medically to try to come up with 200 different treatments for 200 different genetic forms of autism. And really, it's much more than 200. And so one hopes that maybe these different forms of autism might converge into different kind of subclasses or subtypes that might have in common some kind of underlying brain circuit problem that might be different for a few different types rather than 200 different disorders. And so if something like this EI ratio hypothesis is true, it would suggest drug treatments or other interventions that might be developed someday in order to be able to hopefully make an improvement in some whole subclass of autism, rather than having to come up with a different treatment for each genetic type. So that's the motivation. Okay, now I just want to give you an illustration for why thinking about excess spikes as producing noise in some kind of brain computation sort of makes sense intuitively for thinking about how brain function might be altered. And I'm going to give you kind of a toy example here, but it illustrates the question. So this is actually a pictorial representation of how neurons in one region of your brain, which is called somatosensory cortex, which processes information about touch, how those neurons encode a touch stimulus at the fingertip. And this is data that was collected many years ago from primates, actually, in which they're recording the spikes of neurons in somatosensory cortex as raised letters, A, B, C, D, E, are run over the surface of the fingertip. And what you're seeing here is the spike pattern across a bunch of neurons in the brain over time here as these different letters are scanned through. And the basic pattern you can see is that whenever the edge of a letter falls across that region of the fingertip, the cells in the brain whose job it is to encode, did that region on the skin touch something? Those cells spike. The spikes are the dark dots, and so you get this dark density here. And so you can see that in some sense, the ability of the brain to know what letter was there at the fingertip, it depends on the precision of this spike code across neurons and time in the brain. And so if these brain cells were producing excess spikes, those excess spikes would be occurring in other places, other times, and they would blur this. And maybe a blurred code, could be one of the challenges that autistic individuals have to overcome that prevents them from developing normal high-level representation, well, normal sensory representations, or even high representations of things like social interactions or language, communication, etc. So that's the general idea. Okay, so we can study this in mice, and we actually study the somatosensory cortex of mice because it's a very well characterized system where we know exactly how neurons integrate excitation and inhibition and how they decide to spike in a very quantitative way. And we know how that encodes stimuli in the world, kind of like I showed you in that example. So in humans, there's a beautiful map of the body surface in somatosensory cortex. Now, mice and rats, rodents, they also have a whole map of their body surface, but it turns out their most important touch sensor is not their fingertips, it's their whiskers, which they move in back and forth through space. These are mechanosensors that detect vibrations when they hit objects, and they encode for the animal what's around it in the world. And there's a corresponding region of cortex, which is their somatosensory cortex, which I drew a trunk of up here on the top, which we just understand in a lot of detail. And we have a lot of methods in mice to be able to monitor brain activity, for example, by imaging in the brain. I'll show you an example of this on the next slide, or in 2 slides. And then we have access to the function of lots of different cells over here on the right. We can recognize many, many different classes of cells. We know how they're coupled together synaptically. We can measure the properties of all those synapses. We can study how this circuit normally functions. We can study how it functions differently, for example, in genetic models of autism, as I'll show you. And then we can record not only the activity of the network, but we can ask how that network produces behavior, how it regulates itself by changing gene expression, all these things. So there's lots of ways we can study how these circuits are different in genetic models of autism. And my lab has been involved in doing this. Oops, okay, I made a little goof on my slide here, but this is just to kind of schematize one approach we've taken recently to test that EI ratio hypothesis. We can look in that region of the brain in somatosensory cortex, make recordings from different cell types as we study synaptic excitation and synaptic inhibition that converge on those cells to study how they're integrated and whether in fact EI ratio is different in genetic models of autism. And I'm showing you here on the top, I'm just listing a couple of the mouse models of autism, which we have studied, which are very widely studied. There are many models out there. There's probably 15 or 20 good genetic models that genocopy exact human mutations that have been shown to be associated with autism in humans. That is, they confer risk of autism in humans. And when people analyze behaviorally, things like sensory behavior or social behavior in these mice, they show alterations which are reminiscent of some autistic features in humans. So we don't know when we have a perfectly good mouse model. This is one of the big challenges in the field. How do you know if a mouse is autistic or not? But this is a very reasonable guess for mice that have some commonalities with autistic subjects in humans. So here's what one of these we can take little brain slices to make these measurements. Here's what the brain slices look like. We can actually put tiny little glass electrodes inside individual cells to measure synaptic currents and synaptic potentials and how those cells and synapses really work. And we can ask how they differ across these different models of autism. We can also eavesdrop on those neural codes in the brain. If we want to understand, are the codes different in autism? There's many great techniques to do this. I'm going to show you a technique here called 2-photon calcium imaging. This is a technique in which we can genetically express in individual neurons a protein, which is a fluorescent protein that fluoresces when that neuron is electrically active. This protein is called GCAMP6S, which is what this is down here. And here the experiment is, after we express this in these neurons, we can open a small window in the skull of an animal and then cover that up with glass. So the animal now has a cranial window that we can look through to see brain activity. We can put the mouse under a special microscope called a 2-photon microscope, which measures the fluorescence. And in our case, since we want to study how tactile input from the whiskers is represented, we can basically wiggle the whiskers in different patterns as we measure the activity of those neurons to see how they're encoding that somatosensory input. And here's an example of what this brain activity looks like. So you're looking into the brain of a mouse. Each of these little figures here is a cell. There's about 100 neurons in this field of view. They're occasionally flashing. And what we're doing here is we're wiggling different whiskers on the face. That's the flashing box there. And these cells are producing this very sparse spike code where occasionally they will spike in response to one or more of these whiskers, and they flash with the GCAMP signal that we can measure. And by analyzing these types of movies, we can quantify very precisely how hundreds of neurons work together to encode stimuli, what the structure of that neural code is, and then we can compare that across different autism mouse models to ask whether it's different in these models. And there's a little example here on the right of just what this code kind of looks like for different neurons in this region of the brain. Each dot is a neuron in these imaging boxes that are encoding different whiskers on the face. These letter numbers C1, D1, E1, these are different whiskers on the face, and it turns out that different sets of neurons have a job of encoding those different whiskers. Okay, and I just want to give you one example to show you that we can detect differences in how brains encode information in these autism animals, okay? And I'm going to show you, this is actually the one data slide I'll show you. So this is a measurement that's kind of similar to what I just showed you. There's a mouse, whoops, oh shoot, sorry about, my mouse is misbehaving here. So there's a mouse here with a craniotomy over somatosensory cortex. And we're wiggling whiskers to stimulate the brain, to stimulate the sensory input to the brain. And in this case, we're recording not with imaging, but we're actually putting a small electrode into the brain to measure the spikes of neurons directly. And we're going to play in lots of stimuli to these animals. And let me just show you, if you look on the bottom here. So these are plots that show how an individual neuron is encoding stimuli being delivered to 9 different whiskers on the face in a little 3x3 array. So these are 9 different whiskers. There's one whisker here called beta. This is the whisker C1. This is the whisker C2, whisker gamma, D1, D2. This is a little 3x3 row in the face. Each black tick mark here is a spike that we measured in response to deflecting these whiskers. So for example, when we wiggled the D1 whisker, the cell produced lots of spikes right after we wiggled the whisker. But when we wiggled these other whiskers, the cell is just spiking spontaneously. There's really not much evoked activity. And this turns out to be how these cells normally encode whisker input in your average wild type animal. That is, each cell is listening really for inputs coming into one favorite whisker. And this cell in a wild type mouse is just an example of kind of normal coding. And here's a cell that we measured in one of our autism mouse models. This is the FMR1, whoops, that's a typo, FMR1 knockout mouse. This is a model for Fragile X syndrome, which you might know is the most common syndromic form, genetic syndromic form of autism in people. And this individual neuron that I'm picking here responds to its favorite whisker as well, but actually it also responds to this whisker and this whisker and this whisker and that whisker. That is, this cell is kind of has a blurred view of the world. It can't differentiate whether input is coming in from one whisker or a different one or a different one. And this is representative of many cells that we record from in these animals, and this is one of the main phenotypes that we see so far in these autism model mice, that the neural codes seem a bit blurred. And we're trying to understand right now, what's the meaning of that? How does it arise? And what kind of manipulations could we try to develop that might normalize or restore that so it looks more like the wild type case? That's not necessarily something you would want to do in people, but if we knew how to do that, that would give us approaches to think about for therapies in individuals who might want something like that. So that's what I have to share with you guys. Just an overall survey of our approaches.
Thank you, Professor Feldman. It was definitely like very interesting, and yeah, and seeing like using those whiskers to like learn about like different synapses and different like activities and genetic like behaviors. It was just so fascinating and very impressive. And yeah, we saw always in the chat, like many people saying how cool and interesting this is. So thank you again. And so before we enter the Q&A session, I just wanted to, as I know some participants joined us late and may not have seen this in the beginning of the meeting. Our other panelist, Professor Stephen Hinshaw, he is unable to join us today in person because he has an inpatient surgery. And if anyone has questions about his research on stigma in relation to mental and neurodevelopmental conditions, please feel free to contact him through this email or just browse over his lab website. Yeah, so here, now we enter the Q&A time. If anyone has any questions for our two professors, please feel free to speak up or type in the chat.
Hi, I'm Esperanza. I'm going to be moderating the chat for the Q&A. We actually got our first question from Hari, and I believe it's directed towards Professor Feldman. Hari said that this is so interesting to hear about animal models. I have been wondering about the role of the somatosensory, forgive my speech, since childhood. When do you think there will be a move towards convergence, perhaps in the form of precision meds for autistics, especially to address medical comorbidities like seizures, gut dysfunction at a physiological level, also for therapy, for handling sensory overload?
Yeah, that's such an important question. You know, there are many researchers who are rooted in neurobiology who are trying to answer those questions, who are trying to understand. One of the topics you brought up was precision medicine. You know, what are the relationships between genetic mutations in humans and specific types of autism signs or traits? In some cases, it's fairly obvious, like with the strong syndromic disorders. In other cases, it's incredibly subtle and really hard to determine what it is, right? But I think everybody presumes that there will someday be very targeted precision approaches based on the particular form of autism that people have. We're unfortunately not there yet. Now, one challenge actually has to do maybe even with just the somatosensory system. One of the reasons that we focus on somatosensory cortex is that I think the number is 85% of subjects with autism either self-report or their families report sensory disorders, and the sensory disorders are very largely auditory and tactile abnormalities. But those auditory and tactile symptoms, they're not all the same, right? It's not so simple that people are oversensitive or less sensitive, but it's very complex. And so even to understand how that breaks down across different types of autism, I mean segregates, how it segregates across different types of autism, it is a major current research effort and is still unclear. So you ask a great question, but there's an awful lot to do there.
Thank you, Professor, and thank you, Hari, for such a great question. We actually already have another question in the chat. It's by Sylvia. Sylvia says, for Professor Straponi, how does research on language translate to speech therapy? Do you feel like it could be improved?
Thank you for the question, Sylvia. It has changed. For a long time, speech therapists were instructed to try to abate echolalia as quickly and as fully as possible, not recognizing that it could be considered as a resource that can be leveraged to expand the communicative potential of a child with autism. So I think that the recommendation that I would offer to those who work with autistic children in trying to improve their communicative capacities is to first do a thorough analysis of what they deploy already. And my experience in my own research is that by looking at what they are using brings to the surface resources and capacity that was not recognized on a first impression. And while I focus almost exclusively on verbal language, we know that autistic individuals deploy a wide range of communicative resources, not only verbal language, but also they type, they also use and engage with the senses in a way that is very much expressive and offers potentials for intersubjectivity and interconnectedness. So I would recommend to try to understand first the resources that they deploy, both verbal and nonverbal, and try to build on these to certainly try to get the children to expand their communicative repertoire and be able to just have modalities and means to express their being, their identity, their aspirations and their desires in a way that is intelligible, more intelligible and appreciated by neurotypical interlocutors.
Thank you for that response.
Thank you.
Yeah, I actually have another question for Professor Straponi. I'm very interested in the neurodiversity perspective versus the autism perspective you mentioned in the first part of your presentation. I was wondering if you can maybe go a little bit deeper on that, and like how is that specifically reflected on like the methods you use or like the way of analysis you take in your research?
Thanks, Judy. Okay, let me give it a thought, a moment, in order to formulate an answer that hopefully is helpful. Well, one, let me say how I got to feel aligned and to a neurodiversity scholarship. It was not something that I kind of undertook on the basis of ideology or on the basis of a kind of political commitment to promoting diversity and sensibility towards disability studies. I had all of that, but I have to acknowledge that it was the findings of my work that kind of brought me to align and advocate neurodiversity sensibility, because my findings again and again showed resources, capacity that was not previously recognized, and showed also distinctive ways of engaging with the senses that connected the children with the world. So neurodiversity scholars talk about autistic semiosis, that is a distinctive way of engaging the senses, tact, smell, to both explore the world and connected with the world. So in a sense, it's almost ironic that our original understanding of autism was one where the individual was disconnected, detached from the world surrounding them, when it looks like that they are more intimately connected with the world surrounding them through, again, through the senses, first and foremost, and through language as a mode of experience itself. So I didn't get to talk much about another aspect that is fascinating around echolalia. Echolalia often sees children repeating the words of others. And so I've argued that when the children repeat the words of others, and these words can be the words of the teacher, the words of characters from TV, the words of parents, there is an attempt to take the perspective of those. So to put oneself in someone else's shoes by uttering the words of others. So again, here I wanted to suggest that rather than being a meaningless automatic behavior, animating the words of others indicates an attention, a preoccupation towards the other. Perhaps also it indicates a difficulty in taking the perspective of other, what neurotypicals do in a kind of very instinctual, pre-reflexive way. Individuals with autism might need additional kind of efforts and the mediation of language to achieve. But again, in that case, repeating the words of others provides a sensory, acoustic, and almost, articulatory engagement with the other as you produce the words of others. And so in that sense, we can think about this engagement with the senses, with language, as being quite distinctive of autism. And this is what neurodiversity scholars refer as autistic semiosis, and try to bring to our attention and our consciousness, not necessarily as dysfunctional and impairments, but distinctive ways of engaging with others.
Thank you, Professor Starfoni.
Thank you, Professor. You actually have another question in the chat. It's by Renee. Renee says, Dr. Starfoni, do you think of ways that your work on repetitive language can be integrated into physical movements and repetitive sensory experimentation by autistic children as part of interpersonal interactions?
Thank you, Renee. This is thought-provoking, and I feel that probably you have something in mind that can offer ideas, directions, and insights more than I can provide. But the underlying principle and conviction is indeed that there is echolalia and there is echopraxia, so the repetitive movements like steaming, flapping, and those would deserve equal attention. And my hypothesis would be that we could find functionality and potential for communication and engagement with other that we have not explored, but would perhaps bring autistic behavior closer to dance and other spiritual rituals that engage the body in repetitive movements that has not been suggested before. I'm curious to know whether, Renee, you have ideas to offer.
Thank you, Professor, for a great response.
No, I don't have anything in my mind at the moment. I just really enjoyed the talk about mice and their whiskers as sensory processors. And that got me thinking about stimming and the magic of stimming. So yeah, I just feel like that's another thing in the future that could be really great for the research group to think about. Yeah.
Thanks.
Thank you, Renee, for such an enlightening question.
Hi, Professor Feldman. I have a question for you. So I think it's just really interesting to look at that researchers are looking at how to balance the excitatory and inhibitory neurotransmitters when they're trying to find some medical treatment. And I know you mentioned glutamate and GABA, and I've also heard of maybe serotonin that may be involved with autism. So I was wondering if there's any research done on serotonin, and considering how serotonin is usually involved with emotions and mood and learning, would this maybe be related to the anxiety aspect that some autistic individuals may face?
Yeah, that's a really interesting question. There has been study of various neurotransmitters related to autism. I would say that the results haven't been as clear as we might have hoped. So glutamate and GABA, which are the main excitatory and inhibitory transmitters, clearly studied a lot. Oxytocin has been studied a lot, right, for social reward, for example. And serotonin has been studied, but actually often more for the fact that this turns out to be a signaling molecule that is used not only in the brain, but also in the gut. And because of the association with GI problems and autism, people have wondered whether there's maybe some alteration in serotonin production or serotonin use, both in the brain and in the gut. But I don't think there's really been any conclusive proof yet that that's true, but people are absolutely working on it. One of the challenges I have to say that comes up in these biological studies is, let's imagine you could make a measurement of something like serotonin levels, serotonin use, something like that in the brain of an animal that's a good animal model for autism. Just because you can measure those changes in the brain doesn't mean that those changes are causal for the disorder, right? Because this is one of the interesting aspects of neurodiversity, that we all, as animals, as organisms, we're constantly adapting and compensating to our environments all the time. And so by the time you make that measurement in an autism model mouse, maybe what you've measured was an animal's compensation to some other underlying problem and not the problem itself. And this turns out to be a big issue even for the GABA and glutamate questions in autism. So, definitely we can work out these challenges, but this is one of the challenges of studying this in the mouse models. And also, by the way, I think, that's one of the reasons that, one reason I love the neurodiversity idea is that even outside the context of autism, or even outside the context of any, disorder at all, there's simply a wide variety of the way brains work and the way perception works and the way cognition works. And that's because we're all so strongly shaped by this incredible combination, of the lives we've lived, our past experience, and to some extent genes as well. And so brains are really, really different from each other. And so understanding, what aspect of those differences relate to, the traits of, of a diagnosis versus relate to just all of us being individual people, that is a challenging problem.
Thank you, Ellie, for that question, and thank you for that wonderful response, Professor Feldman.
I think we have another question for Professor Feldman in the chat from Hari. Hari says, one issue I think has been that GABA does not cross the BBB, so it has been hard to address imbalances in EI. Often, I think main thing causing this rather intense sensory overstimulation. Thoughts?
Yeah, that's a great point. So Hari, I didn't get a chance to tell you about the main results of my lab's research, but we've actually been studying exactly this EI ratio hypothesis in these mouse models. And many people have studied this in various animal models, and there are even some measurements that can be made in humans a little bit. But you can't really use measurements of GABA or glutamate in blood to be able to make these measurements. And one reason is the one that you brought up, that these don't cross into the blood. But the other reason is that the brain is full of GABA and glutamate, that those pools of those chemicals are not exactly the same pools that are being released and used for neurotransmission. And so even the measurements that people have tried to make of the levels of GABA or glutamate in the brain, like with MRI, with various forms of imaging, that's not really easy to extrapolate to brain function. And when we make these measurements in mice, the bottom line of what we found is there are very prominent changes in EI ratio across multiple different autism mouse models, but they all look like they're compensatory in autism rather than causing the problems in autism. So I'm not convinced that too little inhibition is the problem, personally. It's a big debate in the field right now. And actually, one piece of evidence I would give you is if the problem, in many forms of autism were as simple as too little GABA transmission, too little inhibition, then everybody could normalize themselves by drinking alcohol, because alcohol potentiates responses of the GABA receptors. But that doesn't work, right? So it can't be that simple. It must be something else.
Thank you for that question, Hari, and thank you for that response, Professor Feldman.
Yeah, and Hari also had a question for Professor Sterponi. So we could potentially incorporate and rethink therapy models. And also he added, not think of stings or echolalia as something bad, but as building blocks to coping skills.
Yes, thank you for those questions or suggestions, Harry. And indeed, I think in the spirit of neurodiversity, my orientation is more towards intervening on educational environments for them to be more inclusive of spaces and tools, resources for different ways of engaging with knowledge with each other. So my focus is certainly one that shifts from the individual to the environment. And my own research has shown that there are certain contexts, sequential contexts of language interaction that are more conducive to children's participation and children's production of generative language than others. I've seen that indeed, often children resort to echolalia when they are asked to produce those well-formed sentences, when the parents are kind of turning into therapists themselves, following the ABA directives and asking their children to, again, produce sentences describing things that they have both, under their eyes. That's common knowledge, doesn't need to really be shared verbally. And then in those cases, in a sense, I've observed that when precisely the children are asked to kind of produce scripted language, they revert into echolalia, but not in a way to kind of respond to those pedantic directives, but to resist and divert them possibly towards a more engaging, more playful course of action. And I did see very rewarding episodes where adults kind of trusted the child's capacity to engage in conversation, oftentimes in very playful way, through again those variation into the words of others, playful variation like hot burns, all hot burns, and those sequences saw the children more engaged, initiating more turns and producing also more sophisticated sentences at the end. Once parents kind of went along, gave them trust, rather than imposing their own kind of pedantic therapeutic agenda.
Thank you, Professor. Hari actually has a follow-up to that question. Hari says, so would you consider echolalia like a substitute for the pause sound typical people make, like the ah or or hmm?
I guess, yeah. I haven't seen that in the children that I observed, but it is plausible, and it is a resource that indeed could be mobilized to keeping the turn while thinking and preparing for the production of subsequent strings of thoughts. So it is plausible. I have not empirical evidence, but my data corpus is somewhat limited, and it begs for more research.
Well, it certainly seems like it's opened a door to something really, potential right here. So thank you so much for sharing.
Yes, and thank you everyone for posting those questions. We actually have another question posted by Rachel for Dr. Feldman. Has there been any research done on the neuroscience behind hyperfixations? In my own autistic experience, where I hyperfixate on something for too long, it really feels like my brain quite literally short circuits. So I wouldn't be surprised if any short-circuiting like behavior has been observed in autistic mouse models.
Yeah, that's a very good question. So there are two labs here on campus that are studying the neurobiological basis for this type of phenomenon, which in mice, we can imagine that it's something like cognitive inflexibility or perseveration on a choice task. Like you could imagine you give an animal a choice between object A and object B, maybe one of them is going to give a reward. The animal has to choose, and you construct the task in a way that it benefits the animal to switch around, what it chooses over time. Several of these autism mouse lines, they hyperfixate on kind of the first stimulus that gives them that reward, and they have a very hard time pulling their attention away from that in order to move along in their world. And so these two labs are Helen Badup's lab in the MCB department and Linda Wilbrecht's lab in the psychology department. And they've developed really great tools to try to understand what's happening in the brain that might be causing that hyperfixation. So is it short-circuiting? It's probably not a short circuit in an electrical sense, but there are, mechanisms that the brain uses in order to kind of stay flexible. And maybe some of those mechanisms are a bit more challenging in these autism mouse models. Good question.
Thank you for that question, and thank you for the response to Professor Feldman.
Yeah, and if no one has any additional questions, I just like to thank you again to our panelists, Dr. Starponi and Dr. Feldman, for joining us, and everyone else for joining us for this panel. And yeah, so if you want to stay tuned for with Spectrum, here's our contact info. And yeah, and I think we learned about two totally different perspectives on autism, and we learned about like the social motivations behind echolalia, as well as how the neural circuit has been contributing to like the neurobiological basis for autism. And also we have a lot of inspiring questions in the chat. So yeah, and before we end this meeting, I just like to take a group photo with all our participants. So if everyone is comfortable turning their cameras on, it will be greatly appreciated. Okay, ready? Okay, 3, 2, 1. Thank you, everyone. And I think I will end the meeting here. And thank you, everyone, for being here. And hope you have a great rest of your day.
Okay, nice to meet everybody.
Thank you. Nice to meet you, and keep the good work, Spectrum et al. Keep up with the good work.
Thank you.
Yeah.
Thank you, everyone. What a sigh of relief.
Yeah, that was so good. I missed the first 30 minutes, but it went so well.
Yeah, Professor Serponi actually shared a lot of like extracts of recordings with echolalia, and she like analyzed each of them. So yeah, it was really inspiring, like to hear both.
Good job, Judy and Esperanza. You guys are great moderators. Worked so well
Thank
together.
you. I'm just really glad how impressive our panelists were. Like two completely almost polar opposite disciplines that, yeah, both presentations you could understand.
Right, yeah.
Yeah. I think Dr. Feldman really made like the complicated like neurobiology perspective really understandable for all of us, even like those not majoring in biology.
Yeah. This is what you're going to be learning in 160.
Really? Like, will it be like related to like what he was talking about?
Yeah, there were a lot of concepts like neurotransmitters, and not necessarily autism, but like a lot of the like excitatory and inhibitory like graphs that you saw. It's a lot of neurobio, so you have that coming your way.
Hello, Sylvia.
Hi.
Do you want to turn off the recording?
Oh, yeah.
Yeah, I'm going to turn it off.